I am really excited to be here and share some of the work that we have done. OLAB is interested in early human development, pluripotency, and gene regulation. And today, in the first part, I'm going to tell you about the identification of a chromatin barrier that opposes induction of naive human pluripotent stem cells into alternative cell-fate lineages. And in the second part, I will tell you about a new model for human postimplantation development. In the human embryo, cell fate is gradually restricted. The first cell fate decision segregates the trophectoderm cells, the outside of the embryo, that will become the placenta. And within inner cells, a second cell fate decision takes place that segregates the naive epiblast, from which all embryo cells will come from, and then the primitive endoderm, as you can see here in yellow, which is another extraembryonic lineage. A cavity is formed in the blastocyst, then the embryo implants into the uterus. Other cell-fate decision takes place, including specification of the extraembryonic mesoderm lineage, the amnion, as well as extraembryonic mesoderm. So multiple cell-fate decisions take place, but we still don't understand very much how this all works. In a human blastocyst, things are a little bit different from the mouse. In the mouse blastocyst, just before implantation, all three cell lineages are fully restricted, meaning that cells cannot switch from one lineage to another. However, in the human blastocyst, that's not the case. Trophectoderm cells are not irreversibly restricted. They can still give rise to the naive epiblast, and vice versa. Naive epiblast cells can also still give rise to the trophectoderm, at least in vitro. Several human pluripotent stem cell states have been isolated from human embryos, and also by reprogramming approaches. Primed human pluripotent stem cells represent the post-implantation epiblast state, while naive human pluripotent stem cells reflect the pre-implantation naive epiblast state. And in addition, intermediate pluripotent stem cell states have been isolated in human, such as the formative or extended pluripotent stem cell states. Now, these stem cells offer an opportunity to model development in a dish, and in particular, in the case of human naive pluripotent stem cells, the cells can not only give rise to the prime state that can then give all germ cell lineages, but naive human pluripotent stem cells also have the capacity to give rise to extraembryonic lineages, the trophoblast, as well as the primitive endoderm. And so naive human pluripotent stem cells have complete lineage potential. And this remarkable ability also enables them to self-organize into 3D structures that resemble the human blastocyst, that are termed human blastoids, that have been pioneered by several laboratories, including Nicolas Rivon. So we are now in this exciting time in the field where we have both 2D and 3D human embryo models that can mimic several aspects of human embryogenesis that are normally hard to study in a dish. But in addition, the fact that naive human pluripotent stem cells have complete lineage potential has led to an hypothesis that perhaps the cells are unrestricted epigenetically and devoid of any barriers that oppose alternative cell-fate induction. However, as I'm now going to tell you in the first part, we identified that this is not the case because we found a chromatin barrier that opposes naive to trophoblast fate conversion. So this was an exciting collaboration with multiple laboratories, including the laboratory of Peter Ragan, Henrik Marx, Martin Danens, and also involved Michiel Vermeulen, Nicolas Rivon, and others. And we were interested in understanding how human naive pluripotency is regulated by chromatin processes. And for this, we started by determining the chromatin proteomes, the proteins that are associated with chromatin in naive and human pluripotent stem cells. And we wanted to then understand how chromatin proteins relate to the histone post-translational modifications present in distinct pluripotent stem cell states, and how this in turn affects gene expression program and the developmental potential of naive human pluripotent stem cells. So in order to do this, we've used mass spectrometry approaches and comprehensively determined the chromatin proteins in both naive and prime pluripotent stem cells. These are really rich data sets, thousands of proteins identified, and we can measure the abundance of each of these proteins in both pluripotent stem cell states. And what we see when we compare both states is a lot of changes in the abundance of transcription factors, the key regulators of gene regulatory networks that dictate cell identity, but also many other regulators such as chromatin regulators, writers, readers, and erasers of chromatin modifications, as well as histone variants and chromatin remodelers. So we see a lot of changes. Many of these changes are conserved with mouse, but we also find human-specific changes. For example, a dramatic increase in DNMT3L in the chromatin of naive human pluripotent stem cells. So we then wanted to know how the chromatin proteome relates to histone modifications, and also using mass spectrometry approaches. Sigrid and Martin determined the global abundance of histone modification in naive and prime pluripotent stem cell states. There also we see a lot of changes and a dramatic increase in the histone H3 lysine 27 trimethylation mark in the naive state. So H3 ketone trimethylation is a repressive histone modification that is deposited in chromatin by the polycomb repressive complex 2, PRC2, which is a conserved developmental regulators that's important across vertebrates and invertebrates. And what Peter Ragan's lab found using cat and ran is that H3K27 is not only more enriched in the naive state globally, but also can be found at repressed genes that encode important developmental regulators for alternative cell-fate genes, for example, trophoblast genes such as GATA2. So these results suggested to us that what H3K27 trimethylation might be doing in naive cells is to keep alternative genes of alternative lineages repressed but activatable. And to test that hypothesis, we used an inhibitor of PRC2 that can deplete H3K27 trimethylation. When Irene Talon, a PhD student in our lab, inhibited PRC2, she found that this increased trophoblast fit induction. Therefore, we believe that PRC2 might oppose the induction of alternative cell fate in naive human pluripotent stem cells. And we could also, together with Alok Javali and Nicolas Rivon, test this in human blastoids. And there, what we found is that inhibiting PRC2 in human blastoids also promotes induction of trophoblast fit, but also promote functional attributes of trophoblast. Namely, we saw an increase in cavity formation upon PRC2 inhibition. So in summary, I've told you so far that we have comprehensively identified the chromatin proteins in naive and prime pluripotent stem cells, that we found a marked increase in H3K27 trimethylation in naive cells, and that we found that this functionally opposes induction of alternative cell-fate lineages such as the trophoblast. And we propose that this is one of the ways by which naive cells stabilize the naive pluripotency network against weak pro-differentiation cues during development. But all of the experiments I've told you so far were done in vitro, and therefore we are interested in understanding what happens in the embryo. And excitingly, we've obtained approval to perform similar experiment in the embryo. And we are now, together with Hilde van der Velde in Brussels, testing the effect of PRC2 inhibition directly in human embryos. And the outcome will not only be exciting because it will tell us about the role of polycomb in the embryo itself, but it will also tell us about the predictability of stem cell embryo models on our understanding of actual human embryogenesis. Now, in the second part of the talk, and I want to tell you about a new model of human postimplantation development. There are many challenges when it comes to studying human embryogenesis in post-implantation stages, first of which the embryo is inaccessible for research, and development is not fully concerned with invertebrates. If you just look at morphology, for example, the mouse and human are completely different in post-implantation stages. Also, development is poorly characterized at this stage. We only rely on a few electron microscopy images from decades ago. And the current models we have, be it growing human embryos in a dish to mimic post-implantation embryogenesis, or stem cell embryo models, one of the current limitations is that these do not fully recapitulate the cellular diversity and morphogenesis of the embryo. However, what I'm going to tell you now is that we can now make one additional cell type that's specified just after the embryo implants. I'm showing here an image of about day 14 human post-implantation embryo. What you can see at the outside is a syncytial trophoblast in light orange. Red is the maternal tissue, in dark orange, the cytotrophoblast. In the middle, in yellow, the primitive endoderm, in dark blue, the epiblast. In light blue, the amnion, and in green, all these cells here are the extraembryonic mesoderm. So the extraembryonic mesoderm is an extraembryonic organ that is specified at around day 10 after post-implantation. And the cells then progress to line the inner surface of the cytotrophoblast and primitive endoderm and form a stalk that will act as the primitive umbilical cord. And excitingly, the cells also give rise to the first blood in the embryo, as well as blood vessels. So it's a really important cell type for which very little is known. One of the exciting things that is known is that the timing of extraembryonic mesoderm specification is species-specific. So in human, the cell is specified before gastrulation, but in mouse, the extraembryonic mesoderm is specified from the primitive strate during gastrulation. And so, so these are important differences that we still don't understand. Also, the lineage origin of the extraembryonic mesoderm in the embryo is totally unknown. And importantly, there was no in vitro model for this important human extraembryonic mesoderm cell type. So we initially used a system that was pioneered by other laboratories, including Laurent David, great lab here in France. So it had been described that naive cells can be converted to the trophoblast lineage by culture in trophoblast stem cell conditions. And we did this for 30 days. And as expected and published by others, we found that we could make trophoblast stem cells, large epithelial-like colonies. But unexpectedly, we also had an additional unknown cell type in the culture. And the results were very puzzling at first, but they were consistent. And we could obtain similar results with many different cell lines. Now, in order to identify the identity of these cells, we performed single-cell transcriptomic analysis, analyzed the transcriptome of the cells to align it to a reference atlas of human post-implantation stages. And what we found is that the unknown cells aligned to post-implantation extraembryonic mesoderm from reference human post-implantation atlas from Theiser et al., suggesting that the cells we were making might correspond to extraembryonic mesoderm cells. And indeed, when we looked at the expression of all the known primate-specific extraembryonic mesoderm genes, they were all expressed in those cells. The cells expressed LUM, NIT2, FOXF1, VIM, POST-N, and a unique combination of extraembryonic mesoderm marker that is really unique to the extraembryonic mesoderm. And despite some genes being shared with other cell types, we could really rule out that the cells we were making correspond to additional cell types, such as we could clearly see that they were not amnion or normal mesoderm or any trophoblast derivative or pluripotent stem cells. So these results really suggested that the cells we had isolated correspond to extraembryonic mesoderm cells. I don't want to show you this microscopy immunofluorescent picture of a post-implantation monkey embryo, which was stained with OCT4, marking the epiblast here in red. Here's the amnion, the hypoblast. Outside there's the cytotrophoblast. And in green, all these cells here, marked by VIM, are the extraembryonic mesoderm. And I hope that you can appreciate how extensive this tissue is and how it really embeds the embryo and connects it to the cytotrophoblast. So the point I want to make is that this is not only a beautiful tissue, but this is completely unknown. This is an important cell type for which very little is understood, and all these cells are present in the post-implantation embryo before gastrulation, but that's not the case at all in rodents and in other model organisms. So we verified that the cells we're making from stem cells actually express VIM, and indeed they do, as you can see here in green in these microscopy images of stem cell-derived extraembryonic mesoderm cells. Multiple other hallmarks of extraembryonic mesoderm were recapitulated in our stem cell-derived extraembryonic mesoderm. Extraembryonic mesoderm cells expressed the expected pattern of extracellular matrix genes, one of the functions in the embryo to secrete the extracellular matrix. And also, we could see not only at morphological level, but also at the gene expression level, that the cells undergo an epithelial to mesenchymal transition. And we believe that these might be the cells that undergo the first epithelial to mesenchymal transition of human life. So what else can we learn about these cells? So the key regulators driving cell identity in stem cell-derived extraembryonic mesoderm cells were unknown. And there we used computational tools that can predict key regulators of cell identity. We predicted and reconstructed the gene regulatory networks of pluripotent stem cells, trophoblast stem cells, and extraembryonic mesoderm cells. And these tools correctly predicted the regulatory activity of prime transcription factors in prime cells, naive transcription factors in naive cells, and trophoblast transcription factors in trophoblast stem cells. And then we identified high regulatory activity for transcription factors GETA4, OXA11, OXA10, ARID5B, CREB3L1, and others in extraembryonic mesoderm cells, suggesting that these transcription factors are active and might be functionally implicated in not only inducing but also maintaining extraembryonic mesoderm cell identity, which remains to be functionally tested. The signaling pathways active and required for extraembryonic mesoderm stem cell renewal are also unknown, but we found that both the BMP4 pathway and the mTOR pathway are not only active in extraembryonic mesoderm cells, but are also required for extraembryonic mesoderm cell renewal. And so therefore, this isolation of this new human cell type enables its molecular and cellular analysis. We also wanted to analyze the timing of extraembryonic mesoderm cell specification. And for this, we used time course single-cell RNA-seq analysis, in which we induce naive cells, convert to the trophoblast and extraembryonic mesoderm fate, and then sample the cells at different time points along the differentiation. And what we found, surprisingly, is that naive cells rapidly progress into an intermediate epiblast stage before giving before the appearance of trophoblast stem cells, and at day 8 extraembryonic mesoderm cells that then expand and take over the entire cultures. And so these experiments reveal two things. First, extraembryonic mesoderm cells are specified fairly late, by day 8 in this experiment. And second, they reveal the existence of an intermediate epiblast state. And you may remember that I told you that the lineage origin of the extraembryonic mesoderm in the embryo is totally unknown. And so we try to address this question also in vitro. We induced naive cells to convert, and then sorted or isolated intermediate epiblast cells. And grew them back in culture. And what we found is that intermediate epiblast cells have the capacity to give rise to extraembryonic mesoderm cells, suggesting that intermediate epiblast can, at least in part, give rise to extraembryonic mesoderms, at least in vitro. However, this is still not completely clear. We also found evidence for plasticity because intermediate epiblast cells could also give rise to trophoblast. And when we sorted early trophoblast intermediate, this could give rise to both trophoblast and extraembryonic mesoderm. So we also, I cannot tell you with certainty what the lineage origin of the cells is in vitro. I can certainly not tell you what it is in the embryo. But the interesting hypothesis and suggestion that came from these experiments that it could be that the intermediate epiblast is competent for extraembryonic mesoderm lineage induction, which is an interesting avenue for future research. So I want to finish this part here and summarize. I told you that we discovered a primate-specific property of human naive pluripotent stem cells, the ability to specify and model the extraembryonic mesoderm cell fate. These cells, this system can be used for the molecular and cellular characterization of this important but little understood human cell type. And this will be an exciting new tool for studying primate-specific developmental mechanisms. And so we're really excited by future work using these cells. And our hope is that it might help understand important societal problems, including infertility, miscarriages, and developmental defects, because many of the miscarriages take place at a time when extraembryonic mesoderm cells are specified and present in an embryo. Now, I told you that human blastoids are a great 3D model of human blastocysts, and that they model the 3 founding lineages in a human embryo. But what I didn't tell you is that in all of their published papers, a small subset, and sometimes a large subset, of the cells made in the blastoid assays correspond to off-target cells, cells for which we don't know what they are and that don't correspond to anything that is found in the human blastocyst. So together with Nicolas Rivon and postdoc Bradley Ballaton, re-examined data sets. And what he found is that the blastoids made by Nicolas Rivon, which contain very few off-target cells, only 2%, from these 2% cells, the majority of them actually correspond to extraembryonic mesoderm cells. They express the extraembryonic mesoderm cell gene nature. And so this was a really big surprise. And so Haru Kagawa from Nicolas Rivon went back to blastoids using immunofluorescence to stain blastoids with VIM. And we found that we could indeed find a small number of extraembryonic mesoderm cells restricted to a very few blastoid and non-blastoid structures. And this result could also be repeated in our lab in Leuven. And so it really looks like most blastoids are fine and model the embryo just fine, but in small proportion of cases, there appears to be misspecification of the extraembryonic mesoderm lineage, which should not be present in a pre-implantation model. And we still don't understand why is this happening. So I want to summarize, and I told you that naive human pluripotent stem cells can be used to model early human development. In the first part, we identify chromatin barriers that functionally oppose acquisition of alternative cell fates. And in the second part, we found that the extraembryonic mesoderm can be modeled using naive human pluripotent stem cells. So I now want to quickly tell you a little bit about the future. And our vision is really that we'll be able to recapitulate human post-implantation development in 3D in a dish. This is really an exciting time for the field. We think this will help us understand better development and solve important societal problems. But there are also a number of challenges. And this is that, first, we still don't know how to recapitulate post-implantation human development in 3D. It's still not clear how far we should go. Should we go beyond day 14 with the human embryo? How far can we go with blastoids? Can we go up to, beyond day 14, up to week 4, week 10? what is ethically acceptable? How do we regulate this research? And should stem cell embryo models be regulated like embryos? And finally, how do we communicate with the public? So I want to leave you with that and end by thanking all of the outstanding colleagues I've had the privilege of working with. The work I presented today is the hard work of a really large number of diverse and talented students, postdocs, and outstanding collaborators. And the work that I talk about today was particularly led by multiple people, including Dick Zellmans, Irene Talon, Sigrid Verhels, Adam Bendel, from the Hendrik Marx, Peter Ragan, Martin Danids lab. Also grateful to Nicolas Rivron, Haru Kagawa, and Alok Javali, Laurent David, Alexandre Bruno, many other labs. And then the Extraembryonic mesoderm part was led by Dick Swan-Ifam, Amitesh Panda, and Bradley Balladen. And I want to thank you for your attention.